Welcome to the fertilization module. In this talk, we're going to learn about the processes that occur after the fusion of the male and the female gametes in the context of external fertilization. We will first begin with the formation of a fertilization envelope, followed by the role of calcium in the activation of egg metabolism, and then we will learn about karyogamy. Depending on the localization of the sperm and the egg, fertilization can be external or internal. In the case of external fertilization, the ovum or the egg is laid outside the organism's body. It is usually laid out in the environment, like the sea or a lake or in soil. And in such cases, external fertilization occurs because the process of fertilization is occurring outside the body of the organism. External fertilization is seen in a wide variety of organisms that include sea urchins, frogs, fish, and many other types of invertebrates. It is easier to study the process of fertilization when it is external. And we know a lot about fertilization by studying fertilization in sea urchins. In order for fertilization to occur, the sperm and the egg have to be able to interact. And the first step in this process is chemoattraction, where the sperm is able to follow a trail of molecules secreted by the egg so that it can go close to the egg and find the egg's location. Once the sperm has been able to find the egg, the process of exocytosis occurs where the sperm's acrosomal vesicle is able to release the enzymes that are present in it. These enzymes are able to cleave the different extracellular matrix proteins or other molecules that may be covering the egg so that the sperm can now bind to the vitelline envelope or the zona pellucida of the egg. Once this binding occurs, the sperm has to be able to go through this extracellular matrix to be able to contact the egg cell membrane. Once the sperm is able to contact the egg cell membrane, then the fusion of the egg and the sperm cell membranes can occur. When the plasma membranes of the sperm and the egg fuse, there is a transient increase in the calcium ion concentration in the egg cytoplasm. This in turn results in a cortical granule reaction. The cortical granule reaction results in the formation of a fertilization envelope. So when we look at the figure in the blue box, we're able to see a white-colored egg and many sperm that are surrounding the egg. Where the arrow is shown, that is where one sperm has been able to fuse its plasma membranes with the egg cell plasma membranes. Due to this, we are able to see in that region the formation of the fertilization envelope. The fertilization envelope grows so that it starts covering the entire fertilized egg or the zygote that has been formed, and ultimately we can see that the whole zygote is covered by the fertilization envelope. The fertilization envelope not only protects the zygote, it also helps in preventing polyspermy or other sperm to try to fuse their plasma membranes with the zygote that has been formed. Now let's look a little bit as to how this cortical granule reaction occurs in the first place. And we'll see the importance of calcium ions in this case. Now, calcium ions are released when the sperm enters the egg or even when it contacts the egg. Now, these calcium ions are released from the endoplasmic reticulum, which is where they are normally stored inside a cell. Now, in sea urchins, binding of the sperm to the receptors on the egg cell surface, it will activate the enzyme phospholipase C. So in the picture, in the blue box, we are seeing the interaction of binding with its appropriate receptors on the egg cell surface. And this results in the activation of a membrane-associated protein, which is the phospholipase C. Now, phospholipase C
will cleave one of the phospholipids called phosphatidylinositol 4,5-bisphosphate, which is also called as PIP2. PIP2 gets cleaved to form two second messengers. One is called IP3 and the other is diacylglycerol. And that is what we are seeing in the blue box, where a phospholipid called as phosphatidylinositol 4,5-bisphosphate or PIP2 is cleaved by phospholipase C into two products, diacylglycerol and IP3. Diacylglycerol remains on the membrane, but IP3, which is inositol 1,4,5-trisphosphate, can diffuse into the cytoplasm. Now, both IP3 and diacylglycerol are second messengers. Now, all of this information about the production of IP3 and diacylglycerol should have been covered in the cell signaling part of cell biology. So please do go and read that part if you want to refresh these specific concepts. Now, once IP3 is made, IP3 can go and bind to membrane proteins present on the endoplasmic reticulum membrane, which are ligand-gated voltage channels. So these are basically ligand-gated calcium channels, and when IP3 binds to these channels, the channels open, and calcium that is present in the endoplasmic reticulum can now be released into the cytoplasm. Thus, we can see a local increase in the cytoplasmic concentration of calcium ions. This increase in calcium ions will trigger the cortical reaction to occur. The calcium ions that are released from the IP3-gated calcium channels can promote more calcium channels to open up by binding to calcium-triggered calcium release receptors. And this way we can see that there is an increase in the calcium concentration in the cytosol, and this increase spreads throughout the egg. And this is what is shown in the figures below. So if we look at the first figure that's shown in the blue box, we're actually seeing two egg cells. The one to the left has not yet undergone fertilization, and the one to the right has just undergone fertilization, or the sperm has entered the egg. Now these cells are stained with the dye that will change its color when calcium is present. So when we look at to the egg in the blue box, that is to the right where fertilization has occurred, where we can see this bright white spot, and that is where calcium is now being released. When we look at the second picture now, both eggs have undergone fertilization, and now we can see that in both cases where we see the bright white, that is where calcium is being released. Now the release is occurring to the left side of the egg. Now as we move to the next figure, we can see that in both eggs, now the calcium concentration has increased in such a way or moved in such a way that it is not towards the middle of the egg for the cell that is present on the left, or it is towards the left of the zygote that is present to the right. And thus what we are seeing is a wave of calcium ions that are being released in the zygote. Now, calcium activates metabolic reactions that begin embryonic development, for example, DNA or protein or lipid synthesis. And thus, calcium has many roles to play when it comes to this developmental processes. So, if we summarize all that we talked about now, we can see that the sperm binding and or fusion to the egg cell membrane causes a change in the membrane potential, and this change in membrane potential is a fast block to polyspermy, and thus it prevents more than one sperm fusing with the egg. This fast block to polyspermy is transient, and hence you need a more stable block to polyspermy. Now that sperm binding to the egg cell membrane or fusion with the egg cell membrane also activates phospholipase C. That in turn will synthesize IP3 and diacylglycerol, and that IP3 causes calcium to be released. This calcium that is released will go ahead and initiate the cortical granule reaction. That cortical granule reaction will cause a slow block to polyspermy. In addition, that calcium release can cause or result in many other metabolic changes. So it can result in the restoration of the mitotic cycle, and then now the zygote that is formed can start the process of development, which is the cleavage process where there is rapid cell division. For that, mitosis should occur, and the presence of calcium allows the restoration of mitosis. It also helps in the synthesis of proteins like membrane biosynthesis. The calcium also helps in forming the fertilization envelope. Finally, it stimulates protein synthesis, DNA replication, and cytoplasmic movements of morphogenetic material. Thus, when we see in the entire animal kingdom, calcium plays a very important role in the early development of the zygote. Once the sperm enters the egg, the final step of fertilization requires the fusion of genetic material. For the fusion of genetic material to occur, the sperm nucleus and the centriole separate from the mitochondria and the flagellum. The mitochondria and flagellum of the sperm will disintegrate in the egg cell. And that is why we consider mitochondrial inheritance as maternal. Since the sperm mitochondria are destroyed, the only mitochondria that the zygote has is the one that is present in the egg cell, and hence mitochondrial inheritance is considered maternal. Now, even though the mitochondria disintegrate, however, the centriole that was present in the sperm will persist. And that is why even though mitochondria inheritance is maternal, centriole inheritance is considered as paternal because it comes from the sperm. Now, there are changes that happen to the sperm nucleus. The sperm nucleus tends to be very compact and condensed. And now that it has entered the egg, it decondenses to start opening up, and it forms what is called as the male pronucleus. Now, microtubules extend from the sperm centriole throughout the egg to form an astra-like structure. So when we look at the figure that is shown to the right, all those green thread-like structures that we're seeing are the microtubules that are emanating from the sperm centriole. When these microtubules that are spreading throughout the egg contact the female haploid pronucleus, that is when the male and the female pronucleus can move towards each other. This has been very nicely shown in a series of pictures that have been shown in this specific figure. So in this specific figure, that big circle that you can see a bunch of times is basically the female pronucleus. There's also the male pronucleus and microtubules that are a little harder to see. And so I'm going to show the male pronucleus as a black circle and the female pronucleus as a blue circle, as shown here. Now we are looking at this egg cell at different time periods, which is what are the numbers showing below. So at 0 time point, please observe the location of the female nucleus, which is present as a blue circle, and the male nucleus that is shown in the form of a black circle. Now around the male nucleus we can see the thread-like projections, which are the microtubules that are forming and spreading throughout the zygote. Now we can see that as time passes, the male and the female nucleus start moving closer to each other, and this is normally mediated by those microtubules emanating from the centriole. So with time, we can see the egg nucleus and the sperm nucleus are going closer and closer until finally they fuse right here. And once fusion happens, now you have a zygote. This fusion results in a diploid nucleus, and now the development of the embryo can begin. And with this, we come to the end of the fertilization process that happens in sea urchins.
With this, we come to the end of our talk, where we learned about the formation of the fertilization envelope and the role of calcium in the formation of the fertilization envelope, as well as the activation of egg metabolism. Finally, we learned about karyogamy, which is the process where the genetic material of the male and the female gamete fuse.